This video was developed with help from the Resources for Inclusive Education in Evolution workgroup. For more information and their other available educational tools, check the link in the description. Previous videos in this playlist show the definition, examples, and classification of phenotypic plasticity. In this video, we will see how phenotypic plasticity is measured and the ethical implications of measuring phenotypic plasticity in humans. In the case of transient or acclimation plasticity, it can be easily measured by putting the same individual in different environments and measuring the changes in its phenotypic expression. This is what was done in the study assessing the athlete's aerobic capacity at varying altitudes. Each athlete's VO2 max was measured at different levels of hypoxia, which allowed to estimate the plasticity of this trait in simulated altitude conditions. Developmental plasticity cannot be measured on the same individual because the expressed phenotype is not reversible. However, in organisms capable of clonal reproduction, it is possible to produce multiple clones of the same genotype and expose them to different environments. This is easily achievable in many plants, bacteria, and protists with asexual reproduction, such as the yeast that can reproduce through budding. Since the total phenotypic variation is the sum of genetic variation between individuals and environmental variation which induces the phenotypic plasticity, if we measure the same individual or the same clone genotype, the genetic variation is effectively null, and thus phenotypic variation will correspond to environmental variation. In non-clonal organisms, measuring plasticity requires a more careful experimental design that will control for genetic variation in each of the experimental environments. One way to do this is to use the descendants from the same parents and measure them in different environments. In plants, this is easily achieved by collecting seeds per maternal plant and growing them in contrasting environments. Something similar was done in the fish study that we saw, where guppy fish collected in the ponds were mated in the lab, and then the offspring of each maternal fish was distributed in the two environmental treatments. This approach has its own limitations. Notably, it cannot fully account for the differences between offspring from the same parent due to DNA recombination. But with a solid experimental design, it is easy to disentangle between effects of phenotypic plasticity and genetic differentiation. Quantifying the amount of phenotypic and genetic variation, however, requires controlling for the degree of relatedness between individuals. If you're curious on how this is done, there is a whole playlist on the topic here. The NASA twin study investigated the effect of a year-long stay in space with the help of monozygotic identical twins, the astronauts Scott and Mark Kelly. Both astronauts underwent the same rigorous training, and while Mark was sent on a year-long mission in space, Scott remained on Earth as a control subject. The study measured a vast range of molecular, physiological, and cognitive traits, and showed that most of the measured traits in Mark Kelly returned to the same values as his brother Scott within 6 months after returning to Earth. Some values, such as shortened telomeres and disrupted expression of some genes, did not return to Earth levels by the time the study was published, suggesting potential permanent changes in the DNA structure and its expression. The biggest limitation of this study is obviously its replication size, with only one individual in each tested environment. It is obvious in this case that the main limiting factor is the astronaut training program, whether participants go to space or not, because it is quite expensive, and prolonged stays in space are even more expensive and logistically complicated. Even more affordable experimental designs are struggling to find suitable participants for studies that measure the effects of environmental variation on phenotypic expression. Identical twins are not common, with less than 3 per mil of births being identical twins. Controlling for developmental differences between twins after birth also gets more complicated as they age, with each individual being exposed to different environments based on their personal choices. An infamous example to study the effects of environment on phenotypic plasticity in identical twins was led by child psychiatrist Peter Neubauer and Viola Bernard with the help of the Louise Wise Services Adoption Agency in New York. The study covered more than 2 decades, involved 3 sets of twins and 1 set of triplets, and ended in 1984 with Neubauer's retirement from the Child Development Center. The babies were separated at birth and adopted by families with different socioeconomical backgrounds. The premise for this approach was the now outdated belief that children born of the same pregnancy and placed for adoption would fare better if they were raised by separate families. This presented a unique opportunity to seemingly help orphan babies with a solid experimental design. However, neither the adopted children nor the adopted parents were informed about the existence of other siblings or the actual goal of the study, which is a serious breach of ethical principles in research. Some of the twins or triplets were reunited by chance in their adulthood, which exposed the existence of the study and the lack of fully informed consent from the participants, resulting in several popular media creations telling the story of the twins. The implications and consequences of such studies for the development of research and evolutionary biology will be discussed in the last lecture of this module. For further reading on the topic, check out the links in the description referring to 2 contrasting points of view on the ethics of this study.